Hi, everyone. Thanks so much for coming today. It is my great pleasure to introduce Dr. Angela Garcia. Dr. Garcia is a highly promising fellow in the Center for Evolution and Medicine. She earned her PhD in 2012 from the University of California at Santa Barbara and traded beach sand for desert sand. Her research combines both field work in Honduras and Bolivia, as well as extensive laboratory work. It's rare to find a researcher who's exceptional in both of these very different skill sets. She's published in prestigious journals such as The Lancet, Trends in Genetics, Behavioral and Brain Sciences. Her research is sponsored by the National Institutes of Health, National Science Foundation, Winter Grand Foundation, and the RAND Corporation. More importantly than all of that, she's an excellent collaborator and a wonderful person. Please welcome Dr. Garcia.
Thank you. All right. There's a really profound difference in male versus female clothing, and any probably woman here knows the pockets are a big issue, so they're tiny. Hopefully this doesn't fall out. But thank you guys. Can everyone hear me all right? Okay. Well, so I am happy to be here today and talk to you a little bit about my research, and I'm going to kind of go over two main things I've been working on, so I'll kind of walk you through that. So I'm going to start by giving you a broad overview of the role that your HBA axis, which is your stress response system, and your immune system play in disease risk. And then I'm going to talk about a specific empirical study that I've done looking at going from experiences of psychosocial stress down to risk for metabolic diseases in kind of an ecological context. When I use the term ecologic context, what I mean is a non-laboratory based setting and a less sterile setting than most of the environments in which these kinds of studies are done. Then I'm going to shift into looking at the role of old friends, or parasites, and what the role that they might play in moderating the interaction between your HBA and your immune system. And then I'm going to wrap it all up and talk about the next steps from here. Well, so I probably don't have to tell any of you guys this. Cardiometabolic diseases are a public health crisis. These diseases include things like type 2 diabetes or cardiovascular disease risk. And to give you a little flavor of this, so just in the United States alone, 70% of adults are overweight or obese. And if you look across the globe, 10% of adults, so 1 in 10 people across the globe have type 2 diabetes. And that's actually probably a low ball because there's a lot of, these are all diagnosed cases of type 2 diabetes. And in 1 year alone, in 2017, we spent upwards of, not just we, globally, the globe spent upwards of $727 billion specifically just treating health-related issues related to diabetes. Now, when we think about variation in health and disease or risk for cardiometabolic diseases, we can think about how they're a consequence of these long-term genetic predispositions and responses to particular environments, and also these kind of local socioeconomic and ecological conditions that might trigger or buffer susceptibility to disease risk. For this talk, I'm going to focus specifically on kind of my main thrust of research, which are looking at aspects of how social adversity and environmental exposures interact in relation to stress response systems and immune function in the etiology of disease. So historically, inflammation was considered a major risk factor of all of these metabolic diseases, or cardiometabolic diseases. But there is increasingly this cross-cultural research that is kind of poking some holes in just this one-to-one association between having high inflammation and having high risk for disease. So I'm using, for an example, some research that we've done with the Tsimane, which are an indigenous group from the lowlands of Bolivia. The Tsimane are a mainly subsistence population. They have pretty high pathogen loads. They're pretty active. They have high rates of inflammation across pretty much all the scores here. So this is C-reactive protein, kind of a generalized marker of inflammation. And they're significantly higher than a US-based population at across all ages. Similarly with natural killer cells, which are a marker of an innate inflammation. And also, this is just looking at white blood cell count. And this is in a group of 55 and older. So in this group, about 32% of people were above the cutoff, the US-based cutoff for what would be considered active infection. However, comparatively, the Tsimane have impressively good cardiovasculature. They have almost no heart disease. So I'm going to walk you through this real fast. This is a study that we published in Lancet a couple of years ago. And what we were looking at is comparing Tsimane versus US white populations in coronary arterial calcification. So this is called a CAC score. So if you look at the 50th percentile, you can think of an average US white male that's 80 years old. That person has about a CAC score of 400. Now, a CAC score of 1 means that you have heart disease. A CAC score of over 100 means that you have severe heart disease. You should be going to a cardiologist regularly. You should probably be on statins. So by the time an average white male reaches the age of 80, they have a CAC score of about 400. Now, comparatively, the Tsimane, at that same age, they have a CAC score of 0. And overall, they have almost no metabolic or cardiovascular disease. And the study actually concluded that of all the populations to date that have been studied, the Tsimane have the healthiest hearts. Now, these kinds of diseases, these cardiometabolic diseases, in addition to a lot of other ones, have fallen into this umbrella of what we consider stress-related diseases. What that means is that psychosocial stressors lay somewhere on the pathogenesis of that disease, whether triggering it or exacerbating these diseases. And increasingly, it's been realized that the way this operates is through how psychosocial stress influences the HbA axis and how that then influences the immune system, or leads to a breakdown in the interactions between that HbA axis and the immune system. Now, you probably wonder what the HbA axis is. This is the hypothalamic pituitary adrenal axis. I'm going to talk more about this in a minute. But this is essentially the way that your body physiologically responds to psychosocial stressors. So you experience a psychosocial stressor, and it leads to this stimulation of your HbA axis and a release of a cascade of different hormones and catecholamines. In humans, the main hormone is cortisol. Broadly, we call them glucocorticoids. Now, together, the HbA axis and the immune system regulate the vast majority of your energy in your body. So what you can look at here, this is essentially how your body spends its energy across the lifetime. So on the y-axis, these are kcals over the day that you're consuming, and on the x-axis here is over age. So if you think about, say, the typical undergraduate, maybe you're about 20 years old, all these green bars, or those green lines, those are all the metabolic costs from your body. So your heart, your liver, your kidney, all of those metabolic functions. The purple is the metabolic cost of your brain activity. And the yellow is the immune system. So think about all that together. That's about 80% or more of your energy expenditure is dictated by coordination of the HbA and immune system. So if we want to think about how psychosocial stressors might lead to a disruption in the signaling between these systems, it would probably behoove us to take a step back first and think about how did these systems evolve in the first place. And I'm going to start with the immune system. So this is really tragically oversimplified, but for the sake of this talk, you can think about your immune system as being it lies on a continuum, where on the one side you have the innate immune system, or the innate arm of the immune system, and on the other side you have the adaptive arm. And these systems, or these two arms, kind of work in coordination. The main things to think about here are that your innate immune system, which is comprised of cells like granulocytes, these things are the first responders. They're the first on the scene when you encounter a pathogen. They're a generalized response. They're very effective. They're fast and effective, but the fact they're generalized also means that they cause a lot of collateral damage. So it's kind of like, imagine, there's some sort of issue and you lob a grenade at that issue. You take care of the issue, but you cause a lot of damage in the interim, right? Versus the adaptive immune system. The adaptive immune response is a lot slower. It comes online a few days or a week after you encounter a pathogen, but it's really highly specific. This is like the sniper of your immune system. So it just takes out whatever antigen or pathogen that's encountered, and that's it. It doesn't really cause a lot of other damage. And in particular, though, it's really important for stimulating the factors that are responsible for repairing cells and tissues in your body. Now, Fun fact, and I think this is probably one of the most underappreciated things in biology, in my opinion, 80% or more of your immune system follows a circadian pattern, which means that the levels of your immune cells and signaling molecules actually change throughout the day. And where you tend to have the lowest amount of immunity, or especially adaptive immunity, in the morning, and it increases over the day. And even the relative proportions of immune cells shift. So that you have your innate immune system ramped up in the morning, and that shifts so that your adaptive immune system takes over at night, usually. Now, why would we have a circadian rhythm in our immune system? We can think about this from a couple different perspectives. We can think about it kind of from a life history perspective and a kind of functional biological perspective. And obviously these two are very intertwined. But let's start out with the life history perspective. If you think about over the course of human evolution, we were quite active. Humans have a whole suite of adaptations that are specific to us being active during the daytime, to us being diurnal creatures. So you think about the sun comes up, you're out foraging for food, finding shelter, interacting. This is when you have, a depression of your immune function, but you have relatively more an upregulation of your innate immune system, which makes quite a bit of sense if you think about it, because remember, the innate immune system is the one that's really fast on the scene. That's going to be what deals with these really immediate stressors. Then, the night falls, you kind of you go back to your collective space, you're in closer quarters, there's a lot of kind of close quarter sleeping, etc. There's also some research that's shown that you have an increase in susceptibility to certain diseases, especially human host-to-host past diseases. And you have both an upregulation broadly of your immune function and a shift where your adaptive immune function gets upregulated relative to your innate immune system, which would also make sense if you think about the utility of having those pathogen-specific types of components of your immune system upregulated. And in addition to that, that's also when your body's doing all of its cellular and tissue repair, which would probably behoove you to do when you're not expending a ton of energy on other things, right? To do that while you're resting. Now, from the kind of functional biological approach, these kind of gated oscillatory patterns also minimize the potential of runaway immune responses. So you have, even though I said most of the immune system follows a diurnal pattern, when each cell or signaling molecule peaks is slightly shifted from when the other ones are. And this is really helpful because if you think about if your immune system was just always kind of dialed up to 9 and you experience some sort of pathogenic stressor and everything just freaked out at once, you're going to be probably more likely to have things like sepsis or autoimmune diseases. This is what we see with autoimmune diseases. So now we talked about why we have circadian patterns in our immune system, but how? What regulates those processes? And this is where the HPA axis comes in. So this is the second most coolest and most underappreciated things, I think, is that so most of your cells and signaling molecules, particularly of your adaptive immune system, follow what's called an antiphasic pattern to glucocorticoids. So that means that when cortisol or glucocorticoids are peaking in the morning, that's when your adaptive immune function and a lot of your immunity is at its lowest point. And that diurnal regulatory pattern of your immune cells is actually regulated specifically by or driven by glucocorticoids. So this here shows, this is essentially during the nighttime, you can see that cortisol drops and it leads to a spike in these immune cells, whereas in the morning it spikes and it pushes these immune cells down. This is just an easier way to see the same thing, right? And I'm going to come back to this a couple times. Now, in addition to diurnally regulating your immune function, the HPA axis further optimizes immune responses via this negative feedback loop. So you have a pathogenic stressor that leads to stimulation of your immune system. And then at that point also, though, your immune system is sending signals back to the HPA axis, essentially to tell it, okay, we're good. Well, first regulating it because the glucocorticoids are anti-inflammatory, so it's kind of buffering that inflammatory response. And it helps shut down the inflammatory response when it's time. But in addition to just being kind of having an ecologic adaptation, the HPA axis also has a social adaptation. So humans are really highly social species, right? And there is there's a lot of benefit to having spikes in, or having social situations stimulate your HbA axis. So for instance, if you're doing something like giving a talk, you might have a spike in cortisol, right? But what should happen with that, it should lead to better recollection of salient information. We'll see about that. It should increase social acuity. It should help remember names. In addition, it has a benefit in human social competition. So you guys have probably heard of the fight or flight response. And also, similarly, it has the same, having a stimulation by a social stressor leads to the same immunological shifting of profiles, where it upregulates that innate immunity and it downregulates that adaptive immunity. But this system was likely optimized to respond to relatively acute social stressors. And that's not always the case. What happens when social stressors are more chronic? Now, there have been a lot of studies in mice for everything, but also for chronic social stressors, where they expose this poor mouse to this cat for a long period of time. And even once they remove the cat, the mouse still has essentially a dysregulated HbA system, or a stress response. Now, what can happen when you have these kind of long periods of chronic sustained stress is that your HbA axis stops signaling properly to your immune system. Or another way, actually, a better way to think about this is that your immune system stops responding effectively. And this is called glucocorticoid resistance. So you have these glucocorticoid receptors in the immune cells, and they essentially grab onto the glucocorticoids, and that's what leads to the biological function of the glucocorticoids. But when you have a chronic stimulation of your HbA axis, it can, this is to say that also happens in humans, it can lead to essentially those receptors being unresponsive to the signaling of glucocorticoids. Now, what this is going to look like, I'm going to walk you through actually a graph of what this will look like, what glucocorticoid resistance will look like, because this is what I'm going to get into in the empirical studies. So on the x-axis here, we have the cortisol decline. So think about the decline in cortisol over the course of the day. On the y-axis, we have the change in lymphocytes. So you have the positive change in lymphocytes, or the increase in lymphocytes over the day, right? If you have healthy signaling, you should see an inverse association between these two slopes. So that means that the more shallow of a decline in cortisol, the more shallow of an increase in lymphocytes, right? If you have that, that suggests that your lymphocytes, or the glucocorticoid receptor cells in your lymphocytes, are sensitized to the signals of cortisol. If you don't see that, that's considered glucocorticoid resistance. And now my hypothesis here is that persistent psychosocial stress is going to lead to glucocorticoid resistance. This is not just my hypothesis. This has been something that's quite validated in US and lab-based studies. So quickly, to walk you through the pathway again, just so to make sure that everyone's on board, you have psychosocial stress, hyperstimulating your stress response system, and that leads to a breakdown in communication with your immune system, which is manifested as glucocorticoid resistance, and that leads to higher risk of cardiometabolic diseases. So now let's get to the empirical research. So a lot of my work takes place on the island of Utila. It's an island that's off the coast off of the Honduran coast. There's about 6,000 people that live on Utila, and about 2,000 of them are Honduran immigrants, because Utila is actually, I'm going to use the word founded quite loosely, but founded by British and Americans from back in the 1800s. And I particularly work with Honduran women on the island. This group is overall the most marginalized, both by being Honduran and by gender-based stressors. But there's a lot of variation. They also have high rates of metabolic disease. So a couple years ago, I did an assessment, and in this, Honduran women were actually at the highest risk of any other group on the island. About 60% of women have 3 or more risk factors of metabolic disease. There's 5 risk factors you can have. And if you have 3 or more, it's considered that you have metabolic syndrome. Now, some of the kind of unique things about this island is that there's a lot of socioeconomic variation and variation in exposure to parasites and pathogens. So some people live around the coast here, but a lot of people live in this area called Camponado, which is kind of built into the inland of the island, and it's actually built out over the swamp. And one of the biggest issues here is that there's no sanitation system. So what that means is that there's literally a PVC pipe that runs from the toilet down into the swamp below the house. And that's where all the human refuse, etc., goes. And when it rains, all those swamp waters rise up, and you see these massive spikes in infection rates, etc. The women that I've worked with, this is just a little bit of the kind of demography of them, of this group. So the mean age in the sample is 36 years old. The mean BMI is 29, which is actually overweight. The median income is about $350 US a month, and the median education is about an 8th grade education. So I wanted to see how does psychosocial stress influence associations between diurnal cortisol and changes in immune function, particularly in lymphocytes. Again, this is just to remind you what this is going to look like, healthy signaling versus evidence of glucocorticoid resistance. So I looked at discrimination and depression, and I'm happy to go in more to the details later about the methods, et cetera, that I used, but I'm going to gloss over them right now. And what I found for discrimination was that people that were discriminated against showed evidence of glucocorticoid resistance compared to people that had not experienced discrimination. So again, to walk you through this, this is the slope of cortisol, so the decline of cortisol across the day. And on the y-axis here, we have the diurnal change or the increase in lymphocytes. So people that were non-discriminated, which is this purple line, those people showed healthy signaling between their diurnal cortisol and lymphocytes. People that were discriminated against or that experienced discrimination had almost no association between those two systems. And I found the same thing for people that reported depression. And this isn't just standard day-to-day, like, oh, I don't feel good. This is like, if you're so depressed that it affects your ability to do your daily activities. Now, the next thing I wanted to look at was whether or not glucocorticoid resistance actually does predict metabolic risk. And one of the kind of keys here is that even in US studies or European-based studies, etc., there's very few studies that actually follow this pathway the entire way, from experiencing psychosocial stress to the biological manifestation to disease risk. And what I found was that, indeed, having this evidence of glucocorticoid resistance predicted having higher glucose levels, and having higher risk for having impaired glucose or pre-diabetic glucose levels. So to kind of quickly summarize, the loss of association between HPA and immune function appears to be a pathway through which psychosocial stress can lead to metabolic or cardiometabolic disease risk. Now, when you look across the literature, because this isn't just the case for what I've studied, right? A lot of the studies that are trying to essentially look at how psychosocial stressors are affecting these pathways come up with kind of disparate results. So there's this paradox where, you can see depression that's associated with higher levels of cortisol, but depression is also associated with lower levels of cortisol. Depression is associated with depressed immune function. Oh, but it's also associated with overactive immune function. And I think that one of the problems here is that, one, we're either looking at a single system in isolation, or we're looking at single point or cross-sectional biomarkers and their associations. And the problem with this is, in diurnal systems, depending on kind of what part of the day you're capturing, you're going to see totally different stories, right? Which again is why we should be looking at more than just those cross-sectional associations. Now I'm going to shift to the second part of my talk. So in addition to us, not having a lot of data or understanding of, the effects of psychosocial stress on disease risk in non-US populations, broadly our assumptions about how the social world influences HPA and immune function is in situations that are very different than the mosaic of environments in which we probably evolved, in which these systems evolved, I should say. Because the majority of it comes from these really industrialized societies where we don't have exposure to parasites or pathogens or microbes, etc. And if you think about how those systems evolved, they evolved under conditions where there was probably a history of high pathogen exposure. So I'm kind of trying to transition into understanding what is the role potentially of looking at how exposure to parasites and pathogens could moderate HPA and immune associations. So we have a long history. For the vast majority of human history, we have co-evolved with parasites. And there is a lot of great research done, especially in the archaeology or bioarch, folks that have essentially studied our long history into prehistory of our exposure and host-to-host interactions between us and parasites. What that has led to, or at least this is the kind of a main hypothesis, is that humans have evolved this kind of Th2 biased immune system when you have that background exposure. And you don't have to get too deep into this, but the kind of main difference between Th1 and Th2 responses are that Th2 tend to be biased towards anti-inflammatory responses. And these are the main responses that come online when in the face of a parasite. Right, so we should have kind of this Th2-based response versus this more inflammatory Th1-based response. But there's this other hypothesis that this loss of these kind of old friends or parasites exposure, and specifically the loss of that background immunoregulation from the parasites, has led our systems, our HBA axis and our immune system, to overreact or overrespond to other types of stressors, in particular potentially psychosocial stressors, right? So when you have low background exposure, you experience a stressor and you have this exaggerated inflammatory response. Versus when you have this effective background immunoregulation, you have a relatively normative response, and your system backs down pretty quickly. You guys have probably seen this also in the hygiene hypothesis, which I feel like, it should be rebranded to something more like the biodiversity hypothesis, because we're not saying to not wash your hands. It's more just that when you grow up in an environment where you don't have exposure to a diverse kind of environmental microbes and parasites, then you end up getting kind of this Th1 biased response. So you end up having a more inflammatory-based immune system and relying on inflammatory-based immune responses. What we see, this is what has been linked to the increase in allergies and autoimmune diseases in developed nations. There's also some work that's suggested that this is also one of the reasons that we see an increase of psychiatric pathologies, things like depression in more urban areas. However, there's not a lot of research, again, as I said, on what the HBA axis, the immune system, actually look like and how they associate in high pathogen contexts, because we don't live in them, right? The vast majority of areas where this research is conducted are areas like this, and specifically kind of with undergrads, where we all have pretty low pathogen exposure, right? Except for the anthropologists in the room, which this is actually a photo of Dr. Trumbull here getting infected with the parasite. There's a joke that anthropologists capture parasites the way that most people capture Pokemon, which is probably not untrue. So I wanted to understand, what does parasite exposure influence associations between your HBA axis and your immune system? Now, to capture past exposure to parasites, I'm using the marker immunoglobulin E, or IgE. Oh, sorry about that. So you guys will get that in a second. So there's been a lot of research that has shown, well, first, that when you have an immunological response to a parasite infection, your IgE levels spike. But after the parasite infection is cleared, then the IgE goes back down. But in populations that have pretty chronic parasite exposure, you see an overall increase or overall higher levels of IgE compared to populations that have not experienced parasites. And there's a great paper by our colleague Aaron Blackwell and others that actually looked at tracking levels of IgE with parasitic infections. So this is the Tsimane of Bolivia, who I mentioned, and they have really high parasite loads. About 57% of adults are infected with parasite at any given point. They have the kind of highest level of IgE. The Shuar are a population from Ecuador, and they're also an indigenous population. They do have quite a bit higher parasite loads than the United States. And then this is where Honduras fits here. So about 30% or a little bit over 30% of adults are infected with the type of parasite in the sample that I was working with. And they actually, this is where the 6 times comes in, so the average Honduran woman has an IgE level that's about 6 times more than a US person. So exposure to parasites does modify immune function, but does it influence the interaction between the HPA axis and the immune system? Now, what I found here was that for people that have low IgE, so you have cortisol on your y-axis and the time of day. So people that have low IgE have a pretty steep cortisol decline across the day. People that have high IgE actually have quite a bit more blunted, a significantly more blunted diurnal decline in cortisol over the day. And it's not just the slope, it's actually the peak in cortisol, so that they're not reaching as high of levels and it's not falling as much. And if you include current infection with helminthic parasites in this model, this actually strengthens these relationships. That's what those p-values in the parentheses are. Now looking at lymphocytes and granulocytes, sorry I didn't label, this is the same thing, it's time of day on the x and it's going to be the level on the y-axis. So for lymphocytes, people that have low IgE have a pretty strong increase in lymphocytes over the course of the day. People that have high IgE have a lower increase in lymphocytes across the day, so it's more blunted. For granulocytes, you see no difference between people that have high IgE or low IgE. And also, interestingly, you don't see differences in the base levels of granulocytes or lymphocytes, morning or evening, or the relative ratio of them. But now, how does IgE influence the communication between the HPA axis, or between diurnal cortisol and diurnal immune function? For lymphocytes, you see no change, no difference rather, between people that have high versus low IgE in their correspondence between their cortisol slope and their change in lymphocytes. Interestingly, for granulocytes, that's a different story. So people that have low IgE, they have evidence of glucocorticoid resistance in their granulocytic cell types. So you have almost no association between cortisol slope and diurnal change in granulocytes. However, for people that have high IgE, you have a positive association between cortisol and granulocytes. So to bring this kind of all together, we see that high IgE is associated with lower diurnal decline, or more blunted diurnal change in cortisol, similarly more blunted increase in lymphocytes, but it does not influence the communication between them. And in addition, IgE is associated with differences in association between diurnal cortisol and granulocytes. So what this might suggest then is that without that kind of effective background immunomodulation from parasites, the T1 types of cells, which are mainly the granulocytic cell types, are less sensitive to signaling by cortisol and potentially more prone to dysregulation. And this is an open question that I think that it would be important to explore is, could this explain why we see granulocyte-related diseases like specific types of asthma that are at higher prevalence in low pathogen environments if that system is more prone to dysregulation? Now to kind of bring it back together in conclusion, so we might see variation in traits like immune function or in HPA function that's based on what's optimal or what's useful in a given environment. If you think about the ecological context, essentially giving cues to how those systems should be operating, and in addition, interacting with each other. And also, some aspects of disease might be due to the fact that natural selection has optimized these systems under a really different set of constraints. And so this is the kind of traditional evolutionary mismatch theory where you have these old adaptations that evolved under certain types of constraints and conditions in these novel environments. And then finally, to kind of draw it back to the earlier part of my talk, which I guess this does as well, though, is that something I think that is really underappreciated and very important is understanding the different types of stressors that are triggering the HPA axis and the immune system, and how that is affecting the immune system. Because what this means is that the etiology of certain diseases might be totally different depending on what types of things are leading to that pathology. So you might have, for instance, diabetes, but if even though it's manifesting the same, if the etiology of that disease, if it's triggered based on chronic psychosocial stress influence versus pathogenic stress versus dietary, whatever one of those types of stressors is kind of the largest driver of that might inform then things about how we treat that disease. Now, I started this talk by saying that cardiometabolic diseases are this public health crisis, and they are. And what's worse is that they are rising. They are projected to rise 30% in the next 30 years. And 80% of that is coming from low-income developing countries. Now, why I think this is so critical, and this kind of flavor of research is important, is because understanding what's happening when you have a decrease, so there's a lot of great things that come with development, I should say, right? Access to medical care, medicine, lower infectious rates, but there's also increases in social inequality, increases in potential for exposure to stress, marginalization, etc. Understanding how the decrease in things like parasite exposure, in addition to the increase in things like social inequality and social stressors, both interactively and additively are influencing disease risk, is going to be really important for us to understand how do we effectively intervene earlier, and in general, the types of treatments that we might try to, that what might be most efficacious. All right, with that, thank you guys very much, and I'm happy to take questions.
Thanks so much for such a wonderful talk. So I think we have time for a couple of questions.
Hello. Sorry. Just a question. If long-term stress can induce this desensitizing of the immune system, do you think we can resensitize it by reducing stress or some other means of intervention?
That's a really good question. So I do think that there's been some work to show that, right, if you can modify a person's stress response, then you can have a resensitization of immune cells. And it's still, I mean, it's a standing question, when is too far? And in fact, though, your glucocorticoid receptors over the course of the day even change their sensitivity. And there's also changes in the density and distribution of them and glucocorticoid affinity for them, which means that system is plastic enough, at least up until a point, that modifying things like stressors should be able to impact that. And in addition, we can think about, I mean, drawing it to the second part of the talk, even differences in microbial exposure. And I'm not suggesting that people should, we should not pay attention to discrimination. We should just say, we should just give everyone parasites because maybe that'll help immunomodulate. I'm not saying that. But what I am saying that it is possible that given other kind of exposures, you might be able to resensitize those types of cells. Yeah. That's a good question.
So you showed some examples of how perceived discrimination could influence glucocorticoid resistance. What about other types of psychosocial stressors?
So, yeah, that is a good question. And so far, the work I've done, I have only looked at discrimination, depression, and social isolation. But even with that, and this is actually one of the bigger bummers, is that I found, and I have a slide for this somewhere, so experiencing adversity over multiple domains actually leads to a more severe influence or a more severe risk of glucocorticoid resistance, which again is problematic because if you think about the most marginalized individuals tend to experience stress over a lot of different domains, right? And so it's not just, experiencing something bad versus not, you're at equal risk. There is actually apparently kind of a gradual or an additive effect of multiple stressors. Yeah, that's a good question.
Thanks. In the broader perspective, what you're illustrating on that global map with those huge increases in Africa and much of Asia, really suggests that it's the effect of the second epidemiological transition and the shift to chronic disease as opposed to mortality from infectious disease.
Right.
Just, do you care to comment on that and how that relates to, looking on the smaller scale with the immune functioning and the parasite loads?
Yeah, well, I think that's a great, right, this is, I actually had a slide in here also even, because I was going to use that to make I don't have to show you the epidemiological, well, it's right here anyway, so I'll just put it up. But I think that that's a huge, that is essentially what we would be seeing anyways. Well, I don't want to say anyways, right? Because why we're seeing this at all is still an open question, and it has a lot to do with nutritional transitions, changes in death rates, again, with decreases in infectious disease. But what I think it's in the kind of the interaction between the factors that are leading to this massive increase in disease that is where some of the, even within population, health disparities are really manifesting. And I actually think that one of the things I think a lot about is that even within these transitioning populations, there's still a social gradient and a disease gradient, where you have people that are, even if, population wide, there's an overtaking of chronic or non-communicable diseases relative to infectious diseases, but people that are more marginalized within those areas might be at risk of a dual burden of disease, right? So you have higher exposures still to infectious diseases, and you have exposures to chronic diseases. And I think that that's actually especially troubling, because if you think about when you have things like, glucocorticoid resistance or a dysregulation of your HbA immune that puts you at a higher risk, actually even really functionally, right? If you think about having your lymphocytic cells, for instance, if they're blunted and not signaling properly and not rising in the evening and helping to repair your immune system or your cells and tissues, people that are also experiencing high infectious disease are going to have higher inflammation anyways, which is doing a lot of damage, suggesting that those people that are kind of the marginalized of the marginalized are going to be at greater risk for these at least for the pathogenesis of these diseases. Yeah, I think that that's something I think about so much. It's kind of because you can think about, we can think about global social inequities or social disparities in health, and you can think about how then that manifests on the local levels where you have it's like a tier of who's at the highest risk. Yeah, thanks. I think that's a really important question.
Thanks for that great talk, Angela. I was interested in what you were saying, that we didn't evolve in a chronic stressor environment, right? It was mostly acute stress. So I guess my first part of my question is how much of an acute stress would we have also experienced back when we were hunter-gatherer societies? And two, currently there are animal species that are living under chronic stress, I would imagine. Like if you're a prey animal in the savannah,
yeah,
I imagine they are under chronic stress. So did they evolve any kind of different response than we have?
I love this question. I mean, I guess I love this because I've thought about I've asked myself this a lot. Suggesting that we didn't evolve under chronic stress is actually, we don't know that. It could be, there's been some argument that, again, the domains over which we could experience stress and chronic stress has become kind of more multifactorial throughout human evolutionary history, and that could be an issue. So we are actually experiencing kind of more chronic stress over multiple domains, which we saw increases risk. But it's also, another possibility is that response is adaptive. So having glucocorticoid resistance, for instance, when you experience chronic stress, is not a maladaptation. And if you think about, in fact, when you don't have excessive exposure to calories, there are benefits of being glucocorticoid resistant or insulin resistant. We see this in, for instance, hibernating animals, where bears have put themselves in a space of insulin resistance and glucocorticoid resistance before and actually during when they're hibernating. And it's just kind of a longer, so after, it gets them through that period. But it's an adaptation. And it could be that we see the negative outcomes of all these health consequences in our environment, not just because of chronic stressors, but because in the wild you don't really have access to as many calories that are going to trigger the negative consequences of being glucocorticoid resistant, right? I think that's, and this is again something there's still a lot of research on. What are chronic stressors? Are they are they a novelty? Probably not in some circumstances, right? But maybe over the domains and then exacerbated by things like diet within humans, I should say. And actually, I would love to talk to you about that, whether you see in captive versus wild, because I'm sure that there's probably a difference in calories, right? Yeah.
All right, so I think we have time for one last question.
I can just talk very loudly.
Here, actually.
Like we're recording. I'm wondering if, in your specific population, if perception matters. So if people who perceive themselves to be highly socially isolated or perceive themselves to be highly discriminated versus maybe you just have someone who's alone but very happy.
I don't have it. So this is so silly, and I do not believe in like plugging. I wrote a paper. My master's paper that I published is called "Perception Matters," and I don't have. Of course, I don't have a slide for it. Perception matters. So essentially, what I showed in that paper was that people can experience the same objective socioeconomic or social stressor, but the way that people filter it is totally different. Well, we know that, right, by experience. But so then the question is, does the difference in perception actually map onto biological consequences, or does that have biological consequences? And essentially, people that perceive themselves as having unmet needs, even if objectively they were in the same. So what I did the work on was in socioeconomic space, right? If people had the same socioeconomic status, but they perceived of their status as being worse off, or they didn't have as much as they needed for a sufficient lifestyle, they had much more blunted cortisol over the day than people that were objectively the same and perceived of themselves as better. And that was even controlling for the objective differences in socioeconomic status. And that's also, I think, a huge avenue of research too, is even looking at what drives differences in coping strategies and ability to cope and ability to be resilient. And I've done a little bit of work looking at, for instance, if you experience the same stressor, I was looking at discrimination, but if you have social buffers, so if you feel like you have a strong community and actually feeling strongly a part of your community changed the associations, changed how much that social stressor influenced your biology. I think that stuff is super important. Thank you for that question. That's great.
Alrighty, well, thank you so much for a wonderful talk.
Thank you, guys.